Hi everyone, it is such a pleasure to be here today as part of the ALK Summit again. I wanted to show you that I have my ALK Summit sign from last year too, and I keep it in my office. I really treasure it. Thank you. So we are right now in beautiful Nashville, Tennessee. We loved having parts of the ALK Positive community visit us in February 2020 before the pandemic started. We truly hope that at some point in the not too distant future we can have visitors again. But in the meantime, as part of the summit, we want to show you parts of our lab, and it will be myself and members of my lab team just showing you some of the parts of the work that we do every day and explaining some of the equipment we use and some of the technology we use. So I'm going to start. One question that I frequently get asked is, what does a tumor biopsy look like? And how come there's always a problem with not having enough tumor tissue? So for those of you out there who were here in February, you will remember that we looked at several tumor samples from patients with ALK positive lung cancer, and we went to the special pathology lab at Vanderbilt where those samples are processed. Well, I have some ALK positive tumor biopsies here in my office today, and I'm going to show everybody what they are and explain kind of what the process is. So let's start with an explanation of the process. You all know, because you've all had them, when patients go to have biopsies, a piece of the tumor is removed from the patient. So what happens to the tumor after it's removed from the patient? What's the normal workflow from the time that you have your biopsy to the time that we can actually use it to study your tumor? Well, what happens is a piece of the tumor is taken. It might be a piece of the tumor from the lungs, it may be a tumor in the liver, it may be a tumor in the brain or in the bone. That piece of tumor is taken out of the body and put in something called formalin. Formalin is just a liquid. It looks like water, it's clear, but what the formalin does is it fixes the tumor. So it kind of gives you an instantaneous snapshot of, here's what the tumor looked like when I took it out of the patient. That tumor in the liquid, the tumor in formalin, is then sent to a pathology lab, and it's embedded in something we call paraffin. So you've heard the term formalin fixed, paraffin embedded, or FFPE. You will hear this term all the time, FFPE. That is how tumors are stored, processed, and utilized at any hospital, whether you're in Nashville or New York or Alaska. FFPE, formalin fixed, paraffin embedded, is the standard process for handling tumors. So let me show you a few of those, but pause for a minute because I'm going to put on my mask since this is going to require Ying Jin, who is a wonderful member of my lab, who is videotaping this right now, getting a little bit closer to me. So we're going to practice best social distancing policies and masking. So FFPE, here is one. This sample is actually a tumor biopsy from a patient with ALK positive lung cancer. And what you see here is this cassette. The pink thing is just plastic. If you see here the white stuff, it looks like glue. It's kind of waxy if you feel it. That's the paraffin. So the paraffin is like the glue that the tumor lives in. And then if you look here, and you gotta look really close, I'm gonna zoom in, there's two little specks. There's one right here, and there's one right here. That is a tumor biopsy from this particular patient who had ALK positive lung cancer. So let's just take a moment, let everyone look at that. You see how tiny it is? There's a piece here, there's a piece here. This is tumor that is FFPE, formalin fixed, paraffin embedded, and the paraffin is this white stuff all around it. Okay, let's look at another one. So hold on that one for a second. I have some other examples. So this is a different tumor biopsy. This is also a formalin fixed, paraffin embedded tumor sample. Same cassette, although this one is blue instead of pink. I don't know why we have baby colors today, but that's just the colors we have. There's labeling on the block. You see the paraffin again, the kind of milky white substance. And then here you see this one looks quite different. This is a much bigger tumor sample. I wish every tumor sample looked like this one did, but this one is pretty rare in that it's much larger than what we typically get. And so this one is actually a biopsy from the skin, but all of this brown stuff here, brown, this is all tumor. And so if you look at these two together, these are two different examples of patients with ALK positive lung cancer who have had a biopsy. We have two examples. Here's a tiny little tumor. This is more typical, although this is even smaller than a lot of them I see. And here's a much larger sample. This one's larger than most I see. These are the types of tumors that we can use to send genetic testing from the clinic, to do immunohistochemistry on the tumor samples, or in the lab to isolate DNA or RNA. So I'm going to put these down for a moment. I'm going to zoom out again. I'm going to take my mask off. Okay, it gets stuck in my earring. Now, there's a lot of things we can do with these samples, and we use them for doing genetic testing, for isolating DNA and RNA. We use them for doing immunohistochemistry. We can use them to compare to other patients' tumor biopsies. Let me show you one of those examples. So in this box, I have slides. So this is a bunch of different slides, and there's tumor on every slide. So let's just look at this one, for example. This is a slide of a tumor biopsy, and you can see that it kind of looks pink and purple. How did we get this slide, and what do we use it for? So let's go back to our block for a second. If we took our block, what we can do is shave off pieces of this block. So see how it has a thickness? We shave off pieces, then we take a little piece, a very, very thin piece, usually only 5 microns. That's a thickness that we can't even measure as humans. But we cut it off with a machine, then the tumor piece from the FFPE block goes on the slide, and the slide gets stained with different antibodies or different markers. This pink and purple is something we call H&E, hematoxylin and eosin. And we use H&E to look at the morphology of the tumor, to look at just basically kind of the tumor architecture. And so this is very common, H&E. And so that is one example of the many things that we can do with FFPE tumor biopsies. And as you all know, tissue is always an issue. And so we, in the lung cancer world in general, often get pretty small biopsies, and then we try to study as much as we can from those biopsies, but oftentimes our studies are limited by the fact that we just don't have enough tumor to actually do the things that we want to do. But what I just showed you, FFPE tumor samples, is very standard across any hospital system. That's probably how 99.9% of everybody watching, how their tumor was processed and stored. Last point, you may say, well, what happens to the tumor biopsy after the genetic testing is done? What if there's leftover? So that's a great question, and I will say is these blocks, these formalin-fixed paraffin-embedded blocks of tumor sample, no matter where you are going to be stored in a warehouse or some sort of storage facility for a certain amount of time. So it's possible, and we do this as well, is a patient may have a biopsy, and then a year later we want to look at that biopsy again, and depending on how much tissue is left on the biopsy, we can do different types of studies on older material as well. And so there's a lot of utility in studying FFPE tumors. That is the material we use the most. It is not the only thing we use in the lab, but it is one of them. I am going to let other members of my lab show you different aspects of the work we do and how we study tumors, DNA, RNA, protein, the cells, how we treat the cells with drugs. So stay tuned, those videos will be upcoming. And just before I go, I want to show you the background here. This is the Cellman poster. This is a poster from a patient's wife who we have been in touch with over many years. She comes and visits the lab with her 2 children twice a year, and we just love to have visitors. And when the pandemic ends, we hope that everyone can come and visit us and just really see firsthand what laboratory research is like. We want to thank you all so much for your support, and we're grateful to have this opportunity to partner with the ALK Positive group. Thank you very much, and stay tuned for my lab members coming up showing you some of their cool work.
My name is Henry Henderson, and I'm a current postdoctoral research fellow at Vanderbilt University Medical Center in the lab of Christine Lovely. Today I'm running a Western blot. A Western blot is a method used to detect and analyze specific proteins associated with a particular tissue or cell type. Western blots can also be used to measure the amount of protein expression, for example, ALK in lung cancer. Following separation by electrophoresis, the proteins are then transferred from a gel onto a blotting membrane. Once the transfer is complete, the membrane carries all the protein bands that were originally on the gel. We then perform techniques similar to original photography exposure to visualize our protein of interest. For example, we can confirm the presence of ALK in a lung cancer sample.
Hi, my name is Yun Kai, and I'm a research fellow in Dr. Lovely's lab. Today I'm going to show you an equipment called InQSight, which can bring the new insight of cancer research. Conventional anti-cancer drug research uses an endpoint assay, which measures the cell viability after the drug treatment. These are great assays, but this cannot tell us what the cells look like during the treatment. With InQSight, we can capture the high-definition images of cells repeatedly and automatically. This information will help us to perform real-time quantification of cell behavior to have a better understanding of how the cells react or evolve with drugs, how the drug resistance happens, and how to overcome it.
My name is Juan, and in Dr. Lovely lab, my work is focused on ALK project study. And recently, we are performing a large-scale screening for a chemical inhibitor against ALK fusion resistant cells. And the library contained more than 1,000 compounds. And after screening, we got 18 hits, and of them, 6 hits related to PLK1 oncogene, TPK1, and urokinase A. Additionally, we also have other 8 inhibitors support these hits. PLK1 is an important oncogene related to cell cycle. Related to PLK1 oncogene, P38, TPK1, and urokinase A involved in PLK1 activation. Additionally, according to the publication, PLK1 and TPK1 oncogene knockdown doesn't affect normal cell growth. This is very important for our study. We hope to identify some chemical inhibitors doesn't affect normal cells, but only target cancer cells and resistant cells, especially resistant cells.
My name is Junfang Du. I'm a postdoc in Dr. Lovely's lab. So I'm going to introduce PCR technique to you. PCR is a very common technique to screen DNA mutations. As we know, there are about 22,000 genes within human genome. Based on previous studies, we know non-cancer patients usually have a specific set of gene mutations. We call them hotspot mutations. So to quickly screen gene mutations in non-cancer patients, we can just focus on those hotspot mutations, and the PCR can specifically amplify and select those hotspot mutations out. So usually we collect cancer tissue samples from non-cancer patients and set up a chemical reaction in the PCR machine. And then we start a program in this PCR machine, and we get the PCR product. Then we sequence the PCR product and compare them with normal tissue. Then we will know whether there is a mutation or not in non-cancer patients.
Hi everyone, I hope you're all doing well. First, thank you to all of the patients, all the patients' families, and all the patient advocates who so generously donate and support our research efforts. I am Portia Thomas, an MD PhD student at Meharry Medical College, and I work in the lab of Dr. Christine Lovely at Vanderbilt University Medical Center. Today I'm going to be explaining to you all how to interpret a heat map. And for those of you who don't know what a heat map is, I'll explain. Stay tuned. So today I'm going to explain to you how to interpret a heat map. Now, at first glance, this graph can look extremely daunting, but we're going to go through it piece by piece so that we have a full understanding of what everything on the graph means. So let's first start with the left portion of the graph right here. In this particular graph, and normally on most heat maps, what you will see is a list of something that is being measured on the left side. In this particular case, this thing that's being measured are genes. Now you heard Jim-Fong mention before how the human genome contains over 20,000 genes. So you can see that we're just looking at a very small subset of those genes. On the bottom part of the graph, we have another long list. On this particular graph, this list just happens to be cell lines. This could be cell lines from lung cancer patients. These could be cell lines from patients who have cervical cancer, or they could actually be tumors from those patients as well. In this particular case, these are cell lines that were derived from patients who have small cell lung cancer. The last major thing to point out is, where the heck do all these colors come from? Well, they come from this little bar right here, which is very tiny but means a lot. And what this bar does is shows us in color form the amount of gene being expressed, or otherwise known as gene abundance. So if you look at the numbering, the lower the expression is, or if the expression is 0, then you're going to have these darker, cooler purple, blue colors. And if the expression is higher, then we're going to get into these yellowish colors. And it's just that simple. So within less than a minute, you've now learned how to interpret a heat graph. So what we can do is point out a few things. Where we see all this yellow, we can say that those genes are what? More abundantly expressed. Now this can mean that those genes are being more actively transcribed, or it can mean that for whatever reason, these genes just have a higher expression naturally in these particular cancer cell lines. Then we can look at genes that are not as highly expressed, as noted by these dark blue and purple colors. So it's just that simple. Now you know how to read a heat map. I hope you all learned a lot from that video, and I hope you know now how to interpret a heat map. Take care.
Hi, this is Ying Ji. I'm research assistant in Lovely Lab. Today I'm going to show you cell culture. You may ask what the cell culture is. In general, cell culture means grow cells in lab. In our lab, we grow human non-cancer cells. They are the tools and models for our research projects. The cells I'm going to show you is named STM. It's originally from Cellman, an ALK Positive patient. I guess you have heard the story of Cellman from Dr. Lovely. Dr. Lovely collected the cells and developed it as a cell line, so the cells can continue growing in our lab.
I'm going to show you how we preserve samples so that we can continue our research in a long-term manner. So I'll start with the common refrigerator, that is a household refrigerator equivalent. We have minus 20 in the top, your typical freezer, and there is a 4-degree or a typical refrigerator at the bottom, which is where you keep all your veggies and milk. So we store certain reagents in these two refrigerators, minus 4 degrees and minus 20 degrees. But to do critical research, we need to go beyond sub-0 degrees. So for that, we have a minus 80 degrees centigrade here. And in this one, we can store complex biological samples like DNA, protein, and various forms of DNA and whatnot. So this is minus 80. So compared to minus 20, we're going all the way to minus 80. Here we store samples for years and decades together, and the sample is still preserved and helps us in continuing our research. So beyond minus 80, we need to go much lower so that we can preserve and store certain biological samples like cells and tissues. And this could be stored live. So when we freeze them, we can thaw them back and they can continue to propagate in the lab for us to do more experiments. So this one is called a liquid nitrogen tank. So we use liquid nitrogen to store cells and tissues that we get from patients and from various sources. And this is at minus 180 degrees centigrade. So from minus 80, we are going minus 180 degrees centigrade. So you can see these are the nitrogen vapors, and we have organized storage racks like this, and in each box there are labeled cell vials which contain cells or tissues.
So this really helps us to store precious samples from the clinic and continue our research in a timely manner. Thank you.
Hello. Welcome all ALK Positive patients and advocates. We hope to find you healthy, happy, and most of all, hopeful. Today, from the Vanderbilt University Institute of Chemical Biology High Throughput Screening Facility, also called the Drug Discovery Center, we will be giving you a brief example of the type of work we are doing in collaboration with Dr. Christine Lovely's laboratory in trying to understand ALK-positive lung cancer and how we can discover new treatments.
The Vanderbilt High Throughput Screening Facility, we specialize in taking cells grown in a large flask like this and handling them in smaller plates with 96 wells or even 384 wells. We distribute cells from a single container into a plate with hundreds and hundreds of wells. The goal here is using fewer resources to screen more conditions in a smaller amount of time.
HTS houses around 500,000 unique chemicals that we are able to screen. To store these chemicals, we store them in both tubes as well as multi-well plates. To transfer these tiny amounts of chemicals to add to our cells, we use an acoustic liquid handler, meaning it uses a sound wave to transfer the liquid. Next, we want to add the drugs and chemicals to the cells. We do this by using tips that fit a 384 well head and, like a syringe, pick up the liquid and next transfer it to the cells in the plate. Imaging cells to see what kind of effects these drugs and chemicals have on the tumor cells. So the robot is picking it up from the incubator and is now going to read a barcode to track which plate this is and what type of cells these are. Now it's putting it into a box that contains an automated microscope to image and capture these using a camera. So now with different labels on each cell, we're able to image multiple cells at the same time, or different features of those cells at the same time, using the different colors. So here we have an example of some cells that were treated with a drug that kills off one of the cell types, shown here in orange. And we can overlay these to show how each one of the cells are growing, even in the same well, under the same treatment. And so we can start to understand which cells are more vulnerable to dying by this particular drug than others. Here you can see lots of dead cells with high concentrations of each of the drugs. This can be fully automated to run each of these 384 well plates, and we can load up to 40 2 plates in our incubator, and this can be run and programmed overnight to run 24/7. We can also monitor this through a webcam and control these remotely. And this is how we can test hundreds to thousands to even tens of thousands or 100,000s of different drugs and chemicals to identify new treatments for ALK-positive lung cancer. This is the Vanderbilt high-throughput screening facility.